Okay, good evening, everybody. So I think today all are present. 14 people are there, except apart from me, 13 are there. Okay, so shall we start with the project discussion first? Okay, let me see. Chinmay, can you share your screen? Chinmay, are you there?
Yes, sir.
Yeah, I had sent you some of the links yesterday. Please open that last link, the data flare training. Please open it and kindly share your screen. I am not able to open this. Okay.
Oh, yes, sir. One second. Yes, sir.
Yeah. So, how to give you the groups? Okay, let us do it in this manner. Okay. So, 13 people, 13, that is 12. So, 12, so 3 persons in a group, will it be fine? Chinmay, Alex, Shruti, Prabhu. So let us make it, yeah, okay, yeah, let us make it 3. One person, please note it down. So I was thinking, Chinmay. So everybody is interested in doing the projects, right?
Yes, sir.
Okay.
Yes, sir.
Okay, so what I will do is Chinmay along with 3 persons in a group. So Chinmay, Haripriya and Milan. Okay. Somebody like Alex, can you just type it in the WhatsApp? Yeah.
Okay.
Hello.
Yeah.
Yes, sir.
Yeah. Alex, please type in the chat box. Sorry, in that WhatsApp group. First group, like that is Chinmay, Haripriya, and Geeta. Oh, Geeta, I told Milan. Okay. Then next group, it can be Alex, Geeta, and this is, I think, Priyanka. Which Priyanka? This is Priyanka L. Yeah. So Alex, Geeta, and Priyanka. Is it fine? Alex, are you right? Yeah.
Yes, sir.
Okay, great. So next group, Prabhu, with Rajapriya and Shruti. Where is Pranati? Pranati, she is not interested.
Excuse me, sir.
Yeah.
Yes, sir, sorry to interrupt. Actually, me and Gayatri Pawar wants to do a project together.
You both want to do it together.
Yeah,
Why
sir.
don't you involve one more person?
Yeah,
Make
sir.
it three.
Any more person is fine to us, actually.
Okay, Rajapriya, Gayatri, and you can take Prabhu.
Okay, sir.
Okay. Then Shruti with Shruti, Shailesh. Oh, Shailesh, let him be common with all. Shruti, Yashwanti, and Shravani. Is it okay?
Yes, sir.
Okay.
Yes, sir.
So everybody got allocated. Anybody missed out? I think Chinmay only missed out. Oh, Chinmay,
No,
I
sir.
think
First group.
first group, no? Yeah. So all are there. Anybody whom I left out?
Yes, Sai Divya.
Sai Divya, I told last, no? Last group I told. Sai Divya, Shruti, and Shravani. So Jashwanti is left out.
Sir, 1 second,
Chinmay
sir.
has, yeah.
Sir, myself, Shruti, and Priyanka were planning to do a project, sir.
Yourself, Shruti, and
Priyanka L, sir.
Okay.
okay.
1 second, sir. So let me have a talk. Hello. Hello. I'm audible, right?
Yes, tell me.
Okay. Fine. Okay. So in this batch, who are all the first years?
I have mixed actually. I have mixed everybody.
Yeah, that's
First
what I'm
years.
thinking. So we can mix the second and first year batch.
No, there is no first year here. They are all are second years only, I think.
I mean the output batch.
Outgoing batch and second years.
Yeah. So it will be helpful for the communication.
That is why I kept it in that manner.
Yes.
Yeah.
So here they have mentioned this Rajapriya, Gayatri, and some other person you were saying.
Along with that, I want to give this Yashwanti, because
Okay.
here all are this what you call second year batches.
Okay.
Yes.
This Chinmay is from BTech, right? Okay.
No, one more is see 1 minute. Alex, can you check with once with Praniti?
Okay, sir.
Yeah, then
Yeah,
Pranati and Yashwanti.
Alex, Pranati, and one more work.
No, that's why what you call Pranati and Yashwanti, I will link them. And Priyanka Mukhia. She's no, sorry, even Divya should have joined. So Pranati and Yashwanti, I am trying. So just check out Alex and let me know.
I will, sir.
Yeah, please.
So what's the last group, sir?
Last group is fine. That is Prabhu, Rajapriya, and Gayatri. And last group I want to give. No, I told no Shravani, Shruti, and Sai Divya. Prabhu, I am not considering you because whatever your group is, let it be. Shailesh, is it fine? In all the batches, I included one pass out and.
That's what I'm thinking. So
Yes.
That is good itself only. Why? Because so these people also will learn ongoing batch and these things. So it will be good.
So you and myself have to monitor the students.
Yes.
So anybody who is not present here today, apart from Pranati?
Sir Rushikesh.
Rushikesh is not coming. I have to call him then. I will call him today night and I will see finally. So last batch I was thinking Jovita and Yashwanti. If Praniti is
Divya is
okay,
also
then
there, sir.
So Pranati says
Divya.
she would participate.
Okay, so make it last group. This Pranati, Jovita, Yashwanti, and Divya. 4 people. Okay.
Sir, Priyanka also there, sir. Priyanka Mukia.
Priyanka Mukhya, I will include in some one more group. Okay. I will call her personally. Then I will put Shravani and tell me.
Can you repeat the last group, sir?
Last group is Jovita, Yashwanti, Pranati, and Divya. So I will check out with Priyanka Mukhya and Rishikesh. Then I will confirm. Yes. So Shravani and Sai Divya, is it fine? Shruti?
Yes, sir.
Okay. Okay. Okay, fine. So everybody have known their group. Today even Divya is not here. So let me say I will call her after the class and see what is the plan. Okay. So how many groups are there? 1, 2, 3, 4, and 5. So 5 groups, so 5 data sets. So yeah, now Chinmay, please come down. So first 1 minute. First is the iris. Iris is already used. So come down. Then it is mall customers not needed, not related to your work. What is this? MNEST. Images not needed. Then handwritten digital not needed. Boston housing data set not needed. Okay, next what it is? Data. Okay, fake news detection Python no need. Okay, wine quality. Wine quality, the data set is there. Yeah. So wine quality, one group can take. Which group can take? Chinmay yourself, your group can only take Chinmay, Haripriya, and Milan.
Yes, sir.
Okay, so for you, I am giving this wine quality data set.
Okay, sir.
Yeah. So write it down, somebody. Okay, Milan, just write it. Group 1, wine quality data set.
Okay.
Yeah, just put it in the group.
Yeah, sure.
Okay, Chinmay, please come down. What is this SC over height and weight data set? Yeah, this one you can take it. So height and weight anyway. So the second group, Alex, Geeta, and Priyanka.
Yes, sir.
Yeah, that is Priyanka L. Please inform. So that is group 2. Okay, come down. Next is Parkinson's data set. Yeah, Parkinson data set is Prabhu, Rajapriya, and Gayatri.
Okay, sir.
Sure.
Okay, next is Titanic. Titanic is actually it contains. Okay, you can keep the Titanic dataset on hold. Come down. Come down. Come down. Okay, Uber pics not needed. What is this? CHAR 74. Hindi, Kannada, English, Kannada. That is not related for you. Credit card fraud detection not needed. Come down. What is chatbot? It contains no, not needed. Come down. Next is natural language. That is actually not necessary for you guys. That is natural language. Come down. It is actually for the art student. Aaron emails data set. Okay, come down. What is YELP? What is where did it go? YELP data set. Somewhere I seen. Why it contains. Okay, business attributes. Okay, come down. What is Jeopardy data set? Okay, games not needed. Come down. Come down. The UCI is not needed. Okay. What is the IMBD? It is okay. Sentiment analysis not for you. Come down. MSCO that is also not needed. Flickate. Computer vision. Come down. It is slowly. What is this? Okay, pixels not needed. Come down. German traffic not needed. Image net. Come down. Yeah, this is breast histopathy images. Yeah, keep that one. What is the next group? So Prabhu is done, then Shruti, Sai Divya and Shravani, that is group 4.
Yes, sir.
Okay?
Yes, sir.
So Shruti, Sai Divya and Shravani.
Yes, sir.
Okay.
Sir,
Yes.
Priyanka is waiting, sir, to allow group in Zoom meeting.
Okay. Okay, come down. City space, kinetics, come down. Okay, then MPII. MPII is human data is 40,000 annotated body. Okay, body joints. Okay, that you can take it. So that is the next group you can put. Which is the next group? Yeah, Praniti, Divya. No, this is okay. Praniti, Divya, Jovita, and Yashwanti. That is MPII dataset. Come down. MPII is for this. The Praniti, Divya, Jovita, and Yashwanti. Come down. The photo sketching is not there. I think one more, all are covered.
What's it?
If all the groups are covered, group 5, no, that is the last group. All have got it. Okay, so now Priyanka have joined and Geeta. Geeta, shall I give you project along with Priyanka? Geeta? Geeta, why Geeta is silent? Geeta can drop a chat.
Network issue.
So you can just put it in the chat box. Geeta. So I was thinking for Geeta and Priyanka. Okay, come down. One more data set we have to give. Okay, what is this IMDb wiki? Face images with labeled gender age. Okay, come down. Come down. Color detection, come down. Come down. Come down. What is this? Libre speech not needed. Okay, come down. Come down. Come down. Yeah, what is the data go data? Okay, fine. Come down. Okay, this is health portal data set. Yes. This one, Priyanka can take it, Priyanka and Geeta. Okay?
Okay, sir.
Okay.
Yes,
So,
sir.
yeah, I think in your group I can add her, this Divya. In that group there are 4 people, Divya and Priyanka and Geeta. Okay, so Alex, please change it here. Here it is Praniti, Divya, Jovita, Yashwanti. Just remove Divya and put it in along with Priyanka Mukhia, Geeta and Divya.
Then what about me, sir? I'm in that group.
Which group? You are in the first group, no?
Second group, sir.
You are the second group. There are 2 Priyankas here. One is Priyanka L and other is Priyanka Mukhia. So Priyanka Mukhia with Geeta and Divya.
Okay, sir.
Yeah. So for them you can give this health data portal.
Is it only 2 members in the second group?
2 members in the second group. Why?
Yeah, it's just me and Alex.
Why? Alex, Geeta and Priyanka L. There are 2 Priyanka, one is Priyanka Mukhia and other is Priyanka L.
Yes, but there is only one Geeta.
That's right. Geeta in the last group. Geeta, Priyanka Mukhia and Divya. See here, Divya.
So if Geeta goes to the last group, the second group only will be with 2 people.
Why? Second group there is Priyanka L is there.
Yeah, sir, but there's only one Geeta.
There are 2 Priyanka in the second group. What is the second group I gave? Alex, okay, oh Geeta, okay, Geeta is already there. Oh, okay. Okay. Oh. Anybody who is missing? I think everybody is done, right? Okay. Then okay, last group let it be Priyanka and Priyanka and Divya. And let me see if Rishikesh comes in. Group 6, Priyanka and Priyanka M and Divya. Okay, Geeta in the second group only. Sorry, I got confused. Geeta is there. Okay, I think everybody got the data sets, right? This group did not get last group. Yeah, come down a little bit. Yes, there is this is a Center for Disease Control and Prevention data. This is for the Priyanka and Divya, last group. In the last group, it is Priyanka and Divya. So let me see if any other person I can include. Is it okay, Priyanka?
Okay, sir.
Okay. So everybody, I think got. So please, everybody, create your individual WhatsApp groups. And first thing is that before starting with the project, please try to collect some research papers on the how to do data analysis. Okay, please do not collect the codes first. First, to collect research papers. First to collect research papers, first to study how the research is done, and mainly try to collect the don't see that you are taking papers on R or Python, any language is fine, but try to retrieve all the papers. Even if it is what software is used, please do not consider, because the flow will be the same. So anyway, try to get more papers with R, but if you are not getting also, you can use the other language papers, and after getting it, then only we will start with the project. So what I will do is, from the next class onwards, I will not take it in the weekends. So next week I will not take any class. After that, next week. So there are, I think, 6 groups every day from Monday to Saturday. So group 1 to group 6, every day one person will be coming at evening 8 o'clock. Is it fine? That will be from the which date I will tell you. So from 21st. So 21st, the group 1 will be coming online at 8 o'clock. 22nd, group 2 will be coming online at 8 o'clock. 23rd, group 3 will be coming at 8 o'clock, and so on. 24th, group 4, 25th, group 5, and 26th, group 6. Okay, I want to see what you guys are doing. Instead of taking everybody together, so 3 persons can come online.
Yes, sir.
Okay. Okay. So I am giving you 1 week buffer time, but please do not waste it. Please get all the research papers. Okay, and check it out if at all which all the research is already done on your data set. If you are getting that work also, you can get it the same work. You will not do, you will do something else. Okay.
Okay, sir.
Okay. So I think this Shailesh, please try to next week, please try to come along with me in that 8 o'clock. Shailesh, are you there?
Yeah.
Yeah. Try to come every day online from 21st at 8 o'clock. If you are
Okay.
okay. So any doubt with the group assignment with anybody? Yes or no?
Guys, you are happy with your groups, right? Few people are asking with the partners and also, that's why.
Partners, one person should be at least out so that you can
That's
all
why.
come to know with each other.
Few people were asking, no? Some,
Yeah, Prabhu asked with one person.
Prabhu and some Gayatri, those people are asking.
Yes,
So
yes.
that's what I'm asking. You people are happy, right? The groups.
Yes, sir.
Okay, fine.
Okay. So please, after the class, you can divide your groups and start your work. Start with the initial review of literature. Okay?
Okay, sir. Yes, sir.
Okay. Chinmay, you can unshare your screen.
Yes, sir.
I don't know why Divya did not join today. Okay, so we'll go with the transcriptome restrict. Can be transcriptomics or RNA-seq. Both the names are correct. Same in some of the research papers, you see the name as RNA-seq. sometimes you will be seeing the name as a transcriptomics. So both the names are one and the same. That is why I given the name like transcriptome reconstruction with the RNA-seq. Okay. So generally, how you have to do it? You have to get the RNA-seq data from the sRNA database. Then you have to do experimental and practical consideration, commonly encountered file formats I will show you, and transcriptome analysis methods and the tools. Okay, this is one history. Anyway, I will skip to it because initially how it was done, that how RNA sequence was taken using the various methods. Okay. So I think everybody knows this first step. Here you have the mRNA. So when the mRNA is converted into cDNA, this particular RNA you are taking. Okay, and what happens here is that here you are initially you are breaking the RNA into small fragments. Okay, that is what is your sample. So sample preparation, how it is done? You are breaking your RNA into small fragments. Even it is given here, you have the small fragments here, and it is aligned with the reference sequence. That is the simple basic behind the logic behind this RNA-seq. Okay. So why sequence the RNA? I think everybody knows why we are sequencing mainly the RNA. This is one of the most commonly used method in next generation sequencing. Why mainly RNA? Because RNA is the mediator between your protein and the DNA. So if you are doing with the DNA, it is mainly the whole genome sequencing, and protein it will become the proteomics. So the proteomics study. So sequencing mainly either you can do with the DNA or with the RNA, but DNA sequences are very large. Sometimes your system will get crashed also. So it is always good that you do work with the RNA-seq, that is the transcriptomics. Okay, so differential gene expression you can do it. That is quantitative evaluation and comparison of the transcript levels, usually between the different groups. Vast majority of this is it is actually what you call RNA-seq for differential gene expression. Next is your transcriptome assembly, build or improved profile of the transcribed regions of the genome. That is, which are the transcribed regions which are expressed. That is the transcribed region. And next is meta-transcriptomics, that is mainly for the microorganisms. So transcriptome analysis of a community of different species, that is gut bacteria or host, that is all it is related with the meta-transcriptomics, the term it is related with the microorganisms. So mainly what you are doing here, first is differential gene expression, and then you will be doing the assembly. So what is assembly? I will come to this later. So we are not touching this meta-transcriptomics here. Okay. So usually with the RNA, what you can do, either you can go ahead with the microarray or the RNA-seq. In the RNA-seq, you are preparing the libraries, then you are sequencing the samples, sequencing QC is the quality control, then the reads, and finally you are aligning with the reference genome. So first you are preparing the libraries of the small sequence, then you are checking the quality, then you are aligning with the reference genome. Whereas in the microarray, you need to label the sample, then hybridize the assays, then you have to take the quality of the image. And here you are taking the RNA-seq, you are taking the quality of your particular RNA sequence. And finally, in the microRNA, you have the expression data. The RNA-seq mainly what you are doing, we are checking the quality of the sequence. Here it is mainly the image. Then sample quality control you are doing here, data pre-processing, statistical analysis, and finally you have so many works like Venn diagram, heat maps, annotation and enrichment testing. So here you can see after this is the common in all the this one next generation sequencing. These steps are common till here. From here only it is changing. That is the data pre-processing and statistical analysis for the this one whole genome, RNA-seq and chip-seq or others. It is changing from here. Even with the plant genome, if you are doing the sequencing, till this alignment, it is the same. Okay. So types of RNA, I think everybody knows this one. Ribosomal RNA, responsible for the protein synthesis. It is up to 95% of the total RNA in the cell. Next is your messenger RNA, translated into the protein in ribosome. And you have 3 to 4% of the total RNA in the cell. And they have poly-A tail in the eukaryotes. Then you have the microRNA, that is miRNA. miRNA is mainly used in this one, a small, what is that? RNA interference. RNA interference it is used. So short 22 base pairs of non-coding RNA involved in expression regulation and also in the siRNA, that is the RNA interference. Then you again you have transfer RNA and some of the other RNAs you are here. INC, SH, siRNA is coming here. There is a very little difference between your mRNA. is only short RNA, small RNA. In the siRNA is mainly meant for breaking the DNA. Okay. I know this is small, but it contains the overall of what is your RNA-seq. So first you have the data generation, the total RNA. Then you have to remove the contaminant DNA, fragment the RNA, reverse transcribe the cDNA, then ligate the sequence adapters, then select the range of the size, sequence the cDNA, then you have the data analysis, raw reads, then you are correcting the raw reads which is if needed, and here the main part it is assemble. You are assembling all the reads, the broken reads, and with what you are assembling, you are assembling with the this one reference sequence. So this part actually you are not doing it, but mainly what you are doing here is this part you are doing it. Okay, here it is given in a better manner. That is a data generation. Remove the contaminant DNA, fragment. After fragment, you have reverse transcriptase into cDNA and ligate into the adapter. Actually, this part you are not doing it in the computational part. From here, after fragmentation, you are doing the assembly. So it is here. That is, you are assembling the cDNA ends. Here it is, you are doing it, assembling the cDNA ends. Okay. So how do we sequence the DNA? So from the first generation, way back in 1987, you had the Sagal method. Still it is used sometimes, but it is one of the, is quite expensive and you are getting very less sequence. Then from the second generation, then the so-called next generation word term is used from 2005. Then you have the several other instruments like 454, that is pyro sequencing, solid sequencing by ligation, Illumina. Whenever you are seeing the sequences in your SRA database, you will be seeing it that which particular instrument, with which particular instrument this particular sequencing is done. So we'll have like 454, solid or Illumina or ion torrent or pack bio, and these are the second generation or the next generation. Then third generation, you have this pack bio, nanopore, and 10x. So why I have highlighted here, the maximum sequences are done with this Illumina. Illumina short-read sequencing, rapid involvement over the years between from 36 BP to 300 BP, highest throughput at 100 to 150 BP, many different types of sequencers for the various applications, can also flip a longer DNA strand and sequence from other end to get pair-ended reads. And it is like accuracy of 99.9%. Biases, yes, and most common platform for transcriptome sequencing. That is the Illumina. Okay. So advantages of this RNA-seq. So genome-wide, unlike the microarrays where you look at the selected regions, don't require existing genomic sequence while doing the sequencing actually. So unlike microarray, very low background noise. So I think initially I had told you what is this noise. Noise is mainly the repeated sequences. So reads can be marked with a high confidence or tossed if it is poor quality. Next is the resolution should be of 1 BP. High throughput, much more sequence in a faster time which compared to the Sanger. And cost is, you can see that is most important, 10,000 times cheaper than the Sanger. So that is why now we are trying, Sanger is no more used. Though it was the first sequencer, but still it is not used now because presently with Illumina and others, we are getting the sequencer with around 1,000 times cheaper. Drawback. So everywhere you have to see the drawback also. What is the drawback of this? The depth coverage depends on the sequenceability. That is the GC bias for the PCR based amplification procedures. Okay. So how to get the RNA-seq? So from the SRA database, you can get the RNA-seq. So any person who are interested to do the projects on the RNA-seq, they can carry out within this. They can do this on transcriptomics. Okay. So quality scores estimate the probability of an error in base coding on the quality model. Quality score, it is the for the individual sequence quality model. It includes the quality predictor of a single base, neighboring bases, and the reads. So for a particular read, you are taking the quality model reported for a particular cluster. Okay. And below you have some like some of the characters, like how you can ascertain the particular sequence quality. So you have given some ASCII codes. Do you know what is ASCII code? Everybody? In the FASTQ sequence, you have this ASCII code. It depends on the quality score. It is given here. ASCII quality score. Anybody can tell? Chinmay, please try to open this ASCII in your Wikipedia. Chinmay?
Yes, sir.
Yeah. The ASCII stands for the American Standard Code for International Interchange. So actually the quality, the value will be very high. So instead of giving the so high values, you are giving with the ASCII characters. Yeah, open it. And I want the table. Table is actually beside, it is very small. So try if you can. Okay, first you open the Wikipedia page. Yes, okay, whatever page it is. Yes. So that is the definition. Actually, for the computer people, it is this particular thing is compulsory. They have to study it. Okay.
I'll open it in Wikipedia only.
Yeah, and try to open that box with the ASCII table also. Yes, that table. Not that one, your right hand side, yeah. The second table. Yes, that one. This is the old one. Old table one. For each and every character, you have a ASCII. I think even this is the old table. Okay, fine. Open this Wikipedia page, come down. Yeah. So you can read it. ASCII is abbreviated as American Standard Code for Information Interchange. Is a character encoding standard for the electronic communication. So whenever you are writing some values or some big numbers, it is converted into a particular code, ASCII code. Okay. So come down. This is actually the particular table is a very old. I want to see the new version. Actually, it is okay. New version. Where is the new version table? I'm not getting.
It's not in the image.
It is not given. New version table. Or just try to type in Google the ASCII. I think that table maybe. Okay, open the first table. Yes. This is also, yeah. Still, it is not complete. So you can see the values. So this is actually in the computer they teach it. Decimal, hexadecimal, character. What is this hexadecimal? Anybody can tell? Nobody has idea. Actually, it was a number between 0 to 9, or I think 1 to 10, and you have the letters from A to F. That is hexadecimal. The decimal is 0 to 9. Yeah, it is given here. You can see decimal values 0 to 31, whatever it is. So 0 to 9, it is a hexadecimal. You have 0 to 9, then you have A, B, C, D, E, F. Okay, this is how it is 0 to 9, and hexadecimal is 0 to 9 and A, B, C, D, E, F. And 10, whatever you are seeing, 10 is actually the 0 here. Okay, that is how it is coded here. And see for each and every this one, like this one, space, it is decimal is 20. Then is the exclamation mark. I think. Chinmay, can you little bit enlarge it? Yeah. Yes. Little more. Yes. So you can see here, this is the for the exclamation mark, the decimal value it is 32. This is a double quotes 34, like that. This is a hash. So whenever you are giving any particular symbol in the computer, actually why ASCII is used, you have, so you can see these are the different keywords in your keys in your this one computer or your laptop. So whenever you are giving any particular this one space or what do you call it, exclamation mark, or this one double quotes or single quotes or dollar sign, etc. What happens is that computer, it converts first in the decimal and then actually it converts in the binary. So binary is actually not given here because binary, this particular table cannot take it. It is only given for the decimal. So from the decimal, it is converted to binary because computers cannot read these particular values. That is the decimal, sorry, the characters the computers cannot read. Any character, even ABCD, small or big, it cannot read it. So everything, it has to be transferred in the binary code. So binary is, as you know, it is a combination of 0 and 1. So each and every decimal values, it will get converted into your this one binary. Now, why it is linked it here? Because suppose in the quality score you have, say for example, 10,000. So you have to check it out. 10,000 is matching with which particular character. So actually, in our this one NGS, it is in the reverse manner. So any particular decimal value is there. So this particular decimal value, it is converted into the character value, and it is displayed in the FASTQ sequence. Got it? ASCII table initially was made for character to decimal or binary, but we are using in the NGS as from the decimal value, we are taking the particular character. Okay. Now, Chinmay, please open the FASTQ in this one, Wikipedia. FASTQ in Wikipedia. Then they will understand how it is related. It is just a reverse for your FASTQ.
Sir, is my screen
Yeah.
visible, sir?
Huh? No, your screen is not visible. Yes. Yes, FASTQ, yeah. Okay, come down. Show that. Yes, a little bit up. Yeah, and just maximize is not here. Little bit up you go. Little bit up. Yes, here. From this @ sequence ID. Can you see it? @ sequence ID little bit below. Yeah, @ sequence ID. After this, a FASTQ file containing the single sequence might look like this. So this particular area, please enlarge it. That @ sequence ID, can you see it? Below.
Yes, sir.
Yes. This part, please enlarge it. Yes. So a FASTQ file begins with the @ symbol. Then you have the sequence ID, and below is the sequence. Okay, and then you have the plus sign, and after that you have the ASCII characters. Okay, each ASCII characters denotes a particular quality. So you have to actually, it is we are not actually considering the quality, because we are actually doing the quality checking on each and every symbol. Like it is this first one, you have this exclamation mark. Second one, you have this single dots. Third one also is single dot, a single quote. Fourth one is star sign. Fifth is your first bracket opening. So each of this is denoting a particular value. That is your sequence quality based on the this one sequencer. Based on the sequencer. Okay. Everybody got it? This is how your FASTQ file should look like.
Yes, sir.
Okay. Okay. Please close it. Chinmay, you can unshare the screen. Others, if you have any doubts, you can open the Wikipedia page. Still, if you cannot understand, you can let me know. So that is what is the ASCII quality score. So these are the various sequence formats. FASTA, I am not taking here. FASTA, I think everybody knows. All bioinformatics students know what is FASTA format. Then you have the FASTQ. Then you have the feature formats. One is GFF and GTF. Then you have the alignment format, that is SAM and BAM. Let us see it one by one. So this one already we seen it. Okay, the another is your feature format. So feature format is your GTF and GFF. So used for mapping features against a particular sequence or genome assembly may or may not include the sequence data. Here you may not be able to see the sequence data. The reference sequence, actually this GTF and GFF, yeah, GTF and GFF, it is the reference sequence. It must match with the names of the related file, possibly FASTQ or FASTA. These are versions assembly dependent. They are tied to a specific version, that is assembly release. What is this assembly? Don't take the assembly of your GS step. It is the assembly, that is the sequencer, that is the assembly here of the reference genome. Not all the reference genomes are represented the same. Example, the human chromosome 1. So it will be represented in this manner. So UCSC, you have CHR1 ensemble, you have one NCBI, you will be getting in this manner. So GTF, initially you have the chromosome ID, source, gene feature, so whatever it is, start location, end location, and so on. That is for your GTF. And this is also here. So remember from where you are taking the GTF file. Either you are taking from the ensemble or UCSC or NCBI, it depends. So there will be little difference everywhere. Then finally you have this. One more is GFF. That is a general feature format. So this is little bit different than GFF file. Both the reference genome can be represented either as GTF or GFF. So you can see the difference between the GFF and this is GFF and GTF format. And both the we are using the this one Galaxy can read both. Okay. Now coming to the this one, how we can do this this one sequencing, the steps. First you have to manipulate the FASTQ files. Quality, you have to do the quality control using the FastQC tool or Trigonometrics, either ways. Understand the FastQC output, then use the tools for the quality correction. Okay. So then you have to check it out from where you are taking the sequence. It is always good to take the RNA-seq, otherwise you will not get the results. So you have RNA-seq here, exome-seq. If exome-seq, if you are taking, mainly use it for the whole genome sequencing. ChIP-seq you use only for the chip sequencing experiment. And the DNA-seq and the exome-seq, it is exome and DNA. Actually, DNA-seq data you will not get in the SRA. Not only SRA, anywhere you will not get directly the DNA-seq. You will mainly get the exome-seq. Why? Anybody can tell what is the difference between the DNA-seq and the exome-seq? Anybody? In the DNA, you have both the introns and the exons. And in the exome, you have only the exome data. But problem is, when you are doing sequencing, you will not get the intron data. Usually, most of the sequencers, they will not give you the introns. So it is difficult to get any DNA-seq data. I think Chinmay, I have taken the class for your exome sequencing. I have told you this one. So particular DNA-seq file, it is difficult to get.
Yes, sir.
Yeah. So then it is, you have the quality control, first step of the bioinformatics. So let us do these steps. Everybody, please open your Galaxy. One moment. You please open this. Most of the Galaxy, when I had taken the classes for the Chinmay and others, I was galaxy.org. Then later for your seniors, I used galaxy.eu. Now please use galaxy.be. Everybody please open this use galaxy.ve. There is a Belgium. You always also you have your Galaxy India also, but Galaxy India is tied up with Europe. So Europe Galaxy is not working currently. So that is why you can use this Belgium Galaxy. Can you all see it? Use galaxy.be. I think my screen is shared.
Yes,
sir.
Yes. So what I will do first, I will log out. Why did I log out? I want everybody to register first in the Galaxy. Everybody, I will show you the registration button. That is, yeah, register. Everybody, please register here. You have to enter your email ID, password, public name, that's it. And do it. And then you have to open your email ID. You will be seeing a link. You have to activate it. Do it, everybody, in another 2-3 minutes. If you have any doubts, please ask Chinmay. I will just come and drink water and come. Others, please, Chinmay, please see if everybody is able to register or not. I will just drink water and come. Okay, so for everybody, this Galaxy is activated. For everybody, Galaxy is activated, yes or no?
No, sir.
Okay, do it. NGS you can do in Galaxy. One biggest advantage in here it is you don't have to use the codes. But also using the R you can do sequencing. Maybe in the next workshop I will try to use the NGS using the R. But now you can use this Galaxy only. Okay, everybody's Galaxy is working now.
Yes, sir.
Yes. Yes, sir.
Okay. Okay. So in the chat box, I have given you few datas. One minute, let my Galaxy page open. I will show you. Chinmay, is your Galaxy page open?
Yes, sir.
Can you show them how to?
Yes,
My
sir.
Galaxy is still loading. Yeah.
Yes.
Yeah, show them first. Everybody click on that upload data, then go to this paste fetch data. In the upload data, you see there is a third column, paste fetch data. So you can copy all together and paste it. Then you can start and close it. Start, then you can close it. Got it, everybody?
So could you repeat it, sir?
Okay. Okay, Chinmay, you can share the screen. I will do it.
Okay, sir.
Yeah. Yeah. So first, please open this. Click on this upload data. So first step is you choose from the local files. If you have to upload any data, you can use it. Then there is option called choose FTP files. If you have to take the FTP files and do it, you can use it. Then you have this option called paste fetch data. So this one you can use it. Click on this paste fetch data. Paste all the files which I have given in the chat box and then start. Then you can close it. Got everybody? Yes or no?
Yes, sir.
Yes, sir.
Okay. Okay. Actually, I wanted to show you the quality sequence, quality control today. Let me see if it is, I can show you. Chinmay, is it loaded for you? All the files?
Not yet, sir.
Okay. For the quality control, you have the tool, Fast QC. Just now I showed you. I think it is taking time. Okay, no problem. What you can do, what we will do is we will put this for quality control, quality checking. Okay, tomorrow we can see the results. So tomorrow, please try to come in the morning hours by 9 o'clock. 9 o'clock is fine for you, everybody, morning hours.
Yes, sir.
It's a Sunday,
Yes, sir.
Sunday tomorrow. I
Yes.
think everybody is free. Yes?
Yes, sir.
Yes, sir.
Okay. Okay, please try to come tomorrow morning hours. So between I think 9 to 10, I should complete it. But it is still not loaded. So actually you have to select all the files except this GTF. But it is not showing only except this GTF. Then you have to just give this execute. It's not coming so far. Okay, I think it will take time. Okay, we will meet tomorrow only. I think it is still it is loading. I don't know, it will take some time. Okay, we will see tomorrow at morning hours 9 o'clock. By the time this will load and you will be able to see execute it tomorrow. Okay, and if anybody, and if you can do it on your own, if you are clicking on this multiple data set, you have a single, you have the second one is multiple, and third one is the collection. Click on this multiple, except this GTF file, that is the file number 1. Do not select it, all others you select it and execute it. So try, if you are not able to do it, I will show you tomorrow. So then you can go ahead with the quality checking. In the meantime, you can read about this particular software here. What is the purpose of fast QC, and all these inputs and outputs, everything is given here. Okay? And whenever you are writing any research paper for Galaxy, remember that you have to cite this. You have to use this citation. Okay? So I will wind up here. So I think it will take time. So I will wind up here. Tomorrow morning hours I will take the class morning at 9 o'clock. Okay, see you. Good night.
Good night, sir.
Good night, sir.
Good night, sir.
Good night.
Good night, sir.